Welcome to Farm News, and today I'm with two farmers who made an amazing animal. Welcome, Billy Bob and Joe.
Hello, it's so cool to be here, like I'm so super.
And Joe?
Hello, I'm Joe, and with my sister Billy Bob.
Yeah, I'm Billy Bob.
I think we know that.
What?
Oh, nothing. On to the questions asked by the audience.
Audience, where?
What's the history of genetically modified and transgenic animals?
In my family, 32,000 years ago, I think we had wolves and did magic.
What type of magic?
Magic.
Wait. Our family had bred our East Asian wolves with the
Chinese native dogs. Then people started to be bred more and more doggies.
soon we started to get involved in the DNA.
Who was the first to create a genetically modified animal in your family?
Well, Herbert Janish was the first person.
And Mints, they were the first people to create a genetically modified animal.
I was going to say that.
So they're your family members.
Yep.
Anyways, people want to know, what technology did you use to make this amazing animal?
Computers and DNA technology.
That is very interesting. Also, what are the benefits and risks that could happen in this process?
I'll answer the risks that our animals could get cancer or even worse. I don't want them to die.
And the benefits?
The benefits for Billy Bob's creator is that we get help on how to farm and it make our animals bigger and we can see it in the dark.
Well, one more question before I go.
Oh, we're going already?
After this question,
Oh, I wish you could stay longer.
Billy Bob, you're fine.
on to the question.
The question.
In your opinion, who should be in charge of genetically modified animals?
Us, duh.
No, Billy Bob, it should be scientists.
If they are loyal to their job.
Well, we are done here.
And wait, we have pros and cons.
Okay, but after this, I'm done.
Okay, pros. Farmers can make their crops bigger and make more money.
Cons, their animals could get hurt or they could be diagnosed with cancer or even worse. And we have some demonstrations.
We made this own dark teddy bear.
And once we put a needle in the dog, it made it bigger.
Amazing. Well, after this, I am out. Well, it's time to leave. Bye, folks.
Bye.
Hello, I'm Anissa, attorney-in-law.
Hi, I'm Ava, also attorney-in-law.
Enjoy your information.
Opinion on designer babies.
I think that designer babies can be useful in the future, but also can lead to dangerous side effects. But in my opinion, designer babies is a good discovery.
My opinion about designer babies is that it could be dangerous. The genetic engineers could mess up the genes, but the good thing about it is that it could prevent you from getting cancer or diabetes. I think it is a good and bad discovery.
The first designer baby was made in the 2000s. They took the embryos that would match a baby. IVS and replantation genetic disorders (PGD) are used to create designer babies. Doctors use screening techniques to test the embryos of Lisa and Zach Nash. They chose the embryos that would match exactly to their daughter Molly. Molly suffers from a genetic disorder called fancier anemia, which causes mild deficiency.
CRISPR technology is already widely used by biologists as a kind of search and replace tool to alter DNA, even down to the level of a single letter.
Pros. It increases human lifespan for up to 30 years. It reduces the risk of internal medical conditions such as anemia, obesity, diabetes, cancer, and more.
Cons. Genetic engineers are not perfect people and cannot 100% properly evaluate every gene. Most people will seek out good-looking, intelligent babies with other opportune characteristics. Everyone will be relatively similar.
Designer babies should be regulated. We think that the president should be in charge of designer babies. Also, we think that scientists should be in charge of monitoring it.
Gene Therapy Research Scientist by Carmen and Elena.
Definition of gene therapy. Gene therapy is being researched in clinical studies as a method that attempts to use genes to create or prevent disease.
The transplant of a normal gene into cells is place of missing defective ones in order to correct gene disorder. Question 1A. What is the history of gene therapy and genetic engineering?
The first approved gene therapy clinical research took place on September 14, 1990. One goal of gene therapy being investigated is to introduce functioning genes into the body and try to target the mutations responsible for genetic conditions.
Question 2A. Who helped discover these advancements? September 1990 at the NIH under the direction of William French Anderson.
What were the first type of gene therapy experiments done? 4-year-old Ashani De Silva received a treatment for a genetic defect that left her with ADASCID.
Question B. What type of technology is used in gene therapy? Liposomes medicated transfer involves delivering the rescuing genetic constructed by means of an artificially created lipid barrier called liposomes.
A functional gene is placed inside a transport vehicle, and large numbers are administered via an intravenous infusion.
Question C. What are the benefits and risks of gene therapy? There is not enough evidence about gene therapy as a whole to determine all the possible risks.
Who should be in charge of gene therapy? Scientific organizations should be in charge of regulation and monitoring gene therapy.
The end.
Hi, this is the ACA News, back again with yours truly at Subtract.
Clark Garfunkel.
Andy Kenny.
And today, we found out some interesting things over gene therapy. W. French Anderson is the father of gene therapy. He discovered it. The first experiment on gene therapy was October 19th, 1995, but the first experiment on it was September 1990. Concepts of gene therapy arose in the 60s and the early 70s. The technology used in gene therapy is the gene therapy vectors. These vectors can either be viral or non-viral.
Targeting the wrong cells caused by virus tumors or possibly causing a tumor. Benefits Gene therapy can be designed to a genetic material to cause a concentrate or moral gene or to make benefit protein. And if a mutant gene is causes by necessary protein or faulty or missing gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein.
Scientific organizations should be in charge of regulations and monitoring gene therapy. That could be to make sure nobody is abusing the technology.
Our perspective. We think it is good because people are monitoring it.
It helps get rid of mutated cells.
It is also designed to introduce a genetic material.
And that's the end, folks.
Cloning by Ella and Christopher. History of cloning. Who helped discover cloning? Cloning is the process of making identical copies of an organism, cell, or DNA sequence. First time someone cloned someone is in 1996, and it was on a sheep named Dolly. Number 2. What are the benefits and risks of cloning? The risks of cloning are chance of developmental issues in liver, brain, and heart. It is also very expensive. Due to the benefits are it can prevent diseases. Who should be in charge of regulating and monitoring cloning to make sure that no one is abusing it? Some people would say the government should not regulate cloning because there are too many people that are paying the government to make their own decisions. The FDA or the USDA might be the right person. Number 4. Why cloning is good? We think the reason cloning is good is because we can use it for health benefits such as copy or replicate animals or plants. The end. This is GMOs by Kobe and Nash. Genetically modified organisms. Going by food. The fertilizer helps the food grow. GMO. The interjection is GMOs. The food that is grown are sweet corn and carrots, potatoes and radishes, apples and tomatoes. It goes crops, and then it goes to the stores, sold, and then it goes on your dinner plate. The bigger the crop, the bigger the food. The world is watching what you grow. Will the genetically modified food improve our future? The end.
Hello, we are all professors at Harvard University. We are open to genetic engineering if it has no consequences to humans.
The history of genetic engineering organs. The endotransplantation was the first organ to transplant the first from a different species of animal and people.
Harvard Boyer discovered genetic engineering, and that's why we're talking about this. tech is mostly the main thing is a 3D printer. They use the 3D printer to form or shape the ear, and then they could put it in millions of cells for at least a year. And this is a picture of a 3D printer.
The risks that could happen. What are the risks that could happen? One of the risks is that the organ could get rejected by the body, and the human or animal could get an infection. And the second risk is that the organ that they're trying to replace comes out wrong and could cause pain to the human being. And then the benefits is that it could save millions of people's lives. And the second benefit is that it could be helpful for people that are extremely sick, that are missing an organ or born without one. And who should be in charge? Personally, the government should be in charge. That'd be the best choice. But if whoever the scientist who discovered genetically engineered organs should be in charge.
Or doctors, of course.
Or doctors.
Yep. Scientists or doctors.